Chapter 11 covers meiosis and sexual reproduction. It's not a very long chapter, there are only 2 sections, the process of meiosis and sexual reproduction. The process of meiosis is just discussing fertilization. Fertilization requires the union of 2 different sex cells from 2 different organisms. Meiosis is how we make those sex cells. We look at the polyploidy of organisms, or the number of chromosomes that an organism normally has in a somatic cell. Most animals are diploid. They have 2 sets of chromosomes, and they get 1 homologous chromosome from each partner. So when they create their gametes or they create their spores, they have to have the number of chromosomes in order to undergo reproduction. We refer to this state of having half the number of chromosomes in a gamete or a spore as being haploid. Meiosis follows the same steps as mitosis, but there are 2 divisions and we end up with 4 separate cells that are all different from one another. We refer to these divisions as meiosis 1 and meiosis 2. Each stage has the same name. We just add 1 and 2. So we have prophase 1 and prophase 2, metaphase 1, metaphase 2, so on and so forth. The steps function in the same way and in the same order, just with some differences. The differences allow us to add genetic variation to the potential gametes, which is evolutionarily advantageous. Let's learn the basics of the stages of meiosis, remind ourselves essentially what mitosis looks like, and then expand on some of the specifics. The precursor to meiosis 1 is interphase. Chromosomes are duplicated during interphase. The resulting sister chromatids are held together tightly at the centromere. In prophase 1, the first step of meiosis 1, the chromosomes condense in the nuclear envelope fragments. Those homologous chromosomes are going to bind together firmly all along their length, and we say that they form a tetrad because there are now 4 chromosomes. There's a chiasmata that forms between the non-sister chromosomes, and crossing over occurs at this chiasmata. We'll talk about this in just a second. There are spindle fibers that emerge from the centromeres just as they did in mitosis. In prometaphase 1, the homologous chromosomes attach to the spindle microtubules that are fused with those little kinetochores that are shared by our individual chromatids, just as we learned before. The chromosomes continue to condense and the nuclear envelope completely disappears. In metaphase 1, the homologous chromosomes randomly assemble at the metaphase plate where they had been maneuvered into place by those microtubules that are playing tug-of-war. In anaphase 1, the spindle microtubules pull the homologous chromosomes apart. The sister chromatids are still attached at the centromere. In telophase 1 and cytokinesis, the sister chromatids arrive at the poles of the cells and they begin to decondense. A nuclear envelope forms around each nucleus and the cytoplasm is divided by a cleavage furrow. The result is 2 haploid cells. We say that they're haploid because each cell contains just 1 duplicated copy of each homologous chromosome pair. So yes, they contain 46 chromosomes each, but they're 1 duplicated copy of each homologous chromosome pair, so you don't have that genetic variation that you would find in a normal diploid cell. Thus begins meiosis 2. In prophase 2, those sister chromatids will recondense. They don't always decondense, but in case they do, they will recondense, and a new spindle fiber will start to form. The nuclear envelope once again begins to fragment. In prometaphase 2, the nuclear envelope disappears. The spindle fibers engage the individual kinetochores on those sister chromatids. In metaphase 2, the sister chromatids line up on the metaphase plate. In anaphase 2, the sister chromatids are pulled apart by the shortening of the kinetochore microtubules, and the non-kinetochore microtubules help to lengthen the cell by pushing against one another. In telophase 2 and cytokinesis, the chromosomes arrive at the end of the poles and decondense. Nuclear envelopes surround all 4 of the nuclei. Cleavage furrow forms and the cell divides into 4 haploid cells that are all different from one another. Now let's focus on those chiasmata and crossing over events that lead to the genetic variation. The stages of mitosis and meiosis are largely the same, with some added organization and movement in meiosis. S phase during interphase prior to meiosis produces two identical sister chromatids that are held together by cohesive proteins. This allows different parts of the chromosomes to be switched, and it's kind of like reshuffling a deck of cards together. You still have all the cards you need, just in a different order, and that allows you to be a different deck or to be a different organism out in the wild and have a greater chance for survival. So let's look more closely at those chromosomes. In prophase 1, the homologous chromosomes come together to form a synapse. They're bound by a protein lattice called a synaptomeal complex, and cohesin proteins help to hold them together. Large protein assemblies called recombination nodules direct crossing over events. Crossing over occurs between non-sister chromatids of homologous chromosome. The result is an exchange of genetic material between homologous chromosomes. This random crossing over increases the genetic variation in potential offspring. These chromosomes will be randomly split between sex cells later in the process during metaphase. The random independent assortment during metaphase 1 can be demonstrated by considering a cell with two sets of chromosomes. We would say the ploidy number is 2. In this example, therefore, there are only 4 possible genetic combinations for the gamete. However, in human beings, n equals 23. If you do the math, that means that there are over 8 million possible combinations of maternal and paternal chromosomes, and that's not accounting for the individual genes themselves that may have been crossed over back in prophase 1. The cell cycle moves from metaphase to anaphase just like it did in mitosis. The process of chromosome alignment differs from meiosis 1 and meiosis 2, though. In prometaphase 1, the microtubules attach to the fused kinetochores of the homologous chromosomes. The homologous chromosomes are aligned at the midpoint of the cell in metaphase 1, and in anaphase 1, the homologous chromosomes are separated. In prometaphase 2, though, those microtubules are attaching to the kinetochores of the sister chromatids, and the sister chromatids are aligned at the midpoint of the cell in metaphase 2. Therefore, in anaphase 2, the sister chromatids are separated. This helps to ensure a maximum amount of genetic diversity while making sure that each potential gamete gets the correct number of chromosomes once the process is over. Let's compare and contrast meiosis and mitosis. Meiosis and mitosis are both preceded by one round of DNA replication. However, meiosis includes 2 nuclear divisions. The daughter cells resulting from meiosis are haploid and genetically distinct from one another and the parents. The daughter cells resulting from mitosis, however, are diploid in nature and genetically identical to the parent cell. The second portion of this chapter discussed sexual reproduction. There are 3 major categories of life cycles for eukaryotic organisms that undergo sexual reproduction. They are diploid dominant life cycles, haploid dominant, and an alternation of generations. In animals, sexually reproducing adults form haploid gametes from diploid germ cells. The fusion of gametes gives rise to a fertilized egg referred to as a zygote. The zygote will undergo multiple rounds of mitosis to produce multicellular offspring. The germ cells are generated early in the development of the zygote. At least they are in the case of a female human being, while they're generated over time in males. Fungi, such as black bread mold, have the haploid dominant life cycle. The haploid multicellular stage produces specialized haploid cells by mitosis that fuse to form a diploid zygote. The zygote undergoes meiosis to produce more haploid spores. Each spore gives rise to a haploid organism, again by mitosis. The third and final life cycle for organisms that undergo sexual reproduction consists of an alternation of generation. Plants have a life cycle that alternates between a multicellular haploid organism and a multicellular diploid organism. In some plants, such as the fern seen here, both the haploid and diploid plant stages are free-living. The diploid plant is called a sporophyte because it produces haploid spores via meiosis. The spores develop into multicellular haploid plants called gametophytes because they produce gametes. The gametes of two individuals will form a diploid zygote that becomes a sporophyte eventually. I realize this seems a little out of left field. We haven't talked about animals and reproduction or plants and reproduction as of yet in this textbook, and we'll come across this later on. It's simply to show you how various organisms can undergo meiosis and mitosis to create full-bodied organisms, or just specialized gametes or sex cells that allows them to become different in their environments with each successive generation.